All right, everybody, this video is going to go over proteolysis or breaking down protein. So there's two different ways that we're going to break down amino acids. The first one is transamination. So transamination is basically taking one amino group and transferring it to another amino acid to create a alpha keto acid. So if we're going to use alanine as an example, and we are going to transfer the amino group to another amino acid that we're trying to make, the leftover then becomes pyruvate. So pyruvate is that alpha keto acid. Now, these are catalyzed by two primary enzymes, which are both actually lab values that we see for liver disease. So alanine aminotransferase, or ALA, and aspartate aminotransferase, AST, are the enzymes. So the higher the enzymes are, that means that we're breaking down more proteins, and we might need to increase the amount of protein in our diet to make sure that we're supplying adequate amino acids. So specifically in liver disease, they do require a higher protein diet. So let's actually look at this process. So here we would have our original amino acid, and we're basically going to be transferring that amino group, and it gets reduced to an alpha keto acid. Now here we are going to be transferring that amino group to this other amino acid to basically make an amino acid that is not essential. Now, the second way we can do catabolism of amino acids is deamination, to where we're essentially just removing the amino group. So if you remember, this amino group here on proteins is unique compared to carbohydrates and lipids. So there's no transfer. We simply take that amino acid, we remove the amino group, and it becomes an alpha keto acid. And then what happens is this amino group has to then get rid of in our actual body. So that here would be deamination, where if we continued on and we donated that amino group to an alpha ketoglutarate to become glutamate, then that would be considered a transferase, right? So here we're just getting rid of that amino group, and then we're going to cycle it out through the urea cycle. So what are some fates of amino acids once they are in our amino acid pool? So first, let's look at the energy content of amino acids. They are not optimal for energy. So we are using primarily carbohydrates and fat for energy sources in our body. When we do have a lack of carbohydrates and fat, then we do start breaking down protein for energy as the dominant substrate. However, we're not that efficient. So if you can see, we're going to be putting in some amino acids to pyruvate, and then we can put some amino acids into the TCA cycle or the Krebs cycle. All in all, we do not process amino acids into energy very well in our bodies. So let's look at when we have an amino acid, how we break it down is we split it up into the amino group, which is the toxic side, and then the carbon skeleton. So if we first look at the carbon skeleton, this carbon skeleton can be used for energy. It can be converted to glucose. It can be converted into ketone bodies during starvation. And if we have an excess amount of amino acids in our diet and total calories, then it can be converted to lipids. Now we're going to focus on the next couple slides on how do we actually process this amino group to ammonia to then urea. Now this ammonia, if it's not cycled through this urea cycle, it is toxic. So our body has to get rid of this. So we can measure this with the lab value, blood urea nitrogen. This is the normal range. However, it may vary from site to site, just depending on their actual lab. So they'll give you what the appropriate range is. But overall, this is the appropriate range here. So this is kind of a function, and it measures how well your kidneys are excreting that ammonia in the form of urea. Now, urea cycle enzyme deficiency requires a low protein diet because if not, this ammonia is actually going to build up. And as I said, it can become toxic. So let's look at this step here in the green. So we first are going to have ammonia. And now one thing to note is ultimately we are going to use the TCA cycle. We need 2 ATPs to convert ammonia to carbolamine phosphate. Carbolamine phosphate is then going to become citrulline, and citrulline becomes arginosuccinate. And then arginosuccinate splits into fumarate and arginine. Now, you should recognize fumarate is a step in our Krebs cycle over here, and then arginine is a protein. Now, fumarate can come over and come back into the TCA cycle or the Krebs cycle and continue on. And then arginine becomes ornithine, and ornithine becomes urea, and the leftover part is going to cycle back to carbolamine phosphate. Now, urea is going to be excreted in the urine. So this is why we can test how much nitrogen is in our urine, and so we can look at the protein balance. So let's look at how the TCA cycle assists with this process. So we're going to focus on this step here. So here is a series of using amino acids and the process of using transamination. So we're going to take oxaloacetate and we're going to donate that group to become aspartate. And so we're going to be reducing glutamate to an alpha ketoglutarate. So this is that alpha keto acid. The glutamate is your amino acid. Aspartate then helps combine with citrulline to continue on to arginosuccinate. And then we're going to continue this cycle by transferring that amino group from alanine back to the alpha ketoglutarate to become glutamate again. And then we're left over with pyruvate. So it's this constant cycle so that urea cycle can continue to remove that amino component of the protein. Now, here are some common entry points for amino acids along the different metabolic pathways. So it might be a good idea to be able to match these.